I thought they said they wouldn't use it.
You have to put it on.
Okay, we have today Professor Leon Glass. He's a emeritus professor McGill University in Canada. He has a long career in nonlinear dynamics. He developed the concept of dynamical diseases, and he is going to talk all this history of the dynamical disease concept to us today. And in the title of his conference is Dynamical Diseases, Challenges for Nonlinear Dynamics and Medicine. Thanks, Leon, for being with us.
Should I use the microphone? Okay, so thank you very much. I'm delighted to be here. This is the third conference that I've given, and I'll try to make most of it different from the preceding two. There are a few places where it overlaps. So in terms of disclosures, I do have a contract and had a previous contract and have a license with Medtronic, and I'll mention that in a little bit. And also, I'm not a medical doctor. So in the talk today, I'll first talk about dynamical disease, a little bit of background and history about that, and then I'll talk about some basic science that's been motivated from this concept, and also practical applications of this. So yesterday in the talk that I gave in the Faculty of Science, someone asked, well, what are the practical applications? And in this talk, I'll give more information about some of the practical things that people are talking about that I didn't talk about previously. Now, the term dynamical disease was developed to contrast with the expression genetic disease. If you look on Google Scholar or Google, if you say genetic disease, you get 3 million hits now. The initial start of talking about genetic diseases goes back to 1908, when it was shown that there was Mendelian inheritance of certain diseases of metabolism. Then the famous chemist Linus Pauling demonstrated that sickle cell anemia was associated with a particular abnormal molecule in hemoglobin, and then the actual chemical differences between normal and sickle cell hemoglobin was elucidated in 1956 by someone named Ingrid. In the context of genetic disease, there are many implications for research. People can identify abnormalities, map abnormalities. There's now, of course, the possibility for determining people's genome. The Human Genome Project did this maybe 20 years ago, and now people can get genome analysis from commercial vendors such as 23andMe. It's possible to try to determine abnormalities associated with diseases at multiple loci, and then also to develop therapies. What types of therapies? Well, if you know that you did prenatal testing and you know there's a genetic abnormality, then you could have an abortion, or you could pick a partner who doesn't have a potential of also getting having similar mutations that could give rise to abnormalities associated with recessive genes. Sometimes if you know you have a genetic abnormality that you can't deal with certain nutrients, for example, you can just avoid those. And then increasingly, people have been talking about gene therapy for quite a long time, and now it appears that perhaps at the current stage, with new technologies developed, that there can be gene therapy in which people would increase or decrease expression of genes by replacing or modulating expression of genes. Now, there are many limitations of the concept. We know that many diseases are not just associated with a genetic abnormality, but also environmental factors interacting with that genetic abnormality. There are polygenic diseases that are common and are still not well understood. Gene therapy, as of now, has still been limited. Perhaps it will be much better in the future. People still get sick and must be treated, even if we say, oh, you have a genetic abnormality in your cholesterol and you've had a heart attack. Well, you've still had that heart attack and you have to be treated for that. And disorders often have very large dynamic aspects, even disorders which one might call a genetic disorder. So this is a slide which is a really hard slide to see. This is a slide that was generated by Hobart Ryman, who was a medical doctor who wrote a book which I think is a wonderful book called Periodic Diseases, published in 1963. So it's hard to see what's happening here. This is an axis, just focus over here. This is going from 34 to 40 centimeters. There's a heavy line over here, and there's a dashed line over here. These are oscillating, and they're oscillating out of phase with one another. And the period of oscillation is approximately 1 week. So this is data from a patient. Does anybody have any guess of what is oscillating with a period of a week from 34 to 40 centimeters? Has anybody seen that slide before? Okay, so these are the circumference of the knees of a patient. So Dr. Ryman had a patient, a young lady as it turns out, who had a disease in which her knees alternatively became swollen and not swollen, out of phase with one another for a period of about a week. He got so involved with looking at the knees that he ended up marrying the patient. I met Hobart Ryman at a Gordon research conference that I organized and invited him to in 1977. So I don't know what's wrong. I don't know what that is. If we look at dynamical disease, then instead of 3 million hits, we see 9,000 hits, not very popular. The origin of that term goes back to the summer of 1975. In the summer of 1975, I was in the process of moving from the Department of Physics and Astronomy at the University of Rochester, where I was a postdoctoral fellow and an assistant professor, to McGill, where I was going to the Department of Physiology. In the Aspen Center of Physics, there was a talk by, well, I don't know if Mitchell Feigenbaum gave a talk. There was a talk by Stephen Smale, and he talked about chaos, and he talked about chaos in a quadratic function. So, of course, I did statistical mechanics at the University of Chicago in the Department of Chemistry. The term chaos had not been invented then, and I'm not sure to the extent to which physics students now ever take anything in nonlinear dynamics. So chaos is a term that was initiated around 1975 to describe aperiodic dynamics in deterministic systems with sensitive dependence to initial conditions, and there's also bounded dynamics. So the example that people gave was this nonlinear function, a difference equation. The value of something at one time is a function at a previous time. And if that functional relationship was just given by a parabola, then provided there are certain parameters, certain height of the parameter, you could get a sequence of changes of the dynamics in this difference equation, and you could get what's called period doubling bifurcations, and you could observe chaotic dynamics. This, to me, was an absolutely fascinating concept, having done statistical mechanics where people talk about things that are irregular and aperiodic. I had never heard of this and never realized that something so simple could give rise to these particular properties. When I went back to McGill University, when I started my position, I met Michael Mackey there. Michael Mackey had been trained as a biophysicist and also a mathematician, and he was now in the physiology department. And I asked him, I just learned about chaos. Can physiological systems be chaotic? Is it possible that we can find this in physiological systems? And he said, I don't know. Maybe we should start thinking about that. So what we did is we started to develop mathematical models of nonlinear systems which were motivated by the way physiological systems work. Now, this is from a paper that I showed yesterday. This is from a paper we wrote then in the summer of 1977, in which we have, this is experimental data of blood cell dynamics, having very striking dynamics, and this is a mathematical model that we developed. The mathematical model says that the rate of change of blood cells is a function of their production minus their destruction. And for the production rate, we were motivated by some of these early discoveries of difference equations where if one has a non-monotonic function, one can observe chaotic dynamics. In this case, the non-monotonic function can be justified physiologically by saying if there are a lot of blood cells, you don't need many. If there are very few blood cells, you can't produce many because you're sick. So there's some intermediate region. You have some maximal production of blood cells based on some simple feedback system. But there's always a time delay in this system, so that time delay has to be taken into consideration. So we had a nonlinear time delay differential equation in which we observed chaotic dynamics, and we also, in that paper, mentioned the concept of dynamical diseases. And the notion was that this was going to occur as a result of a bifurcation, a change in dynamics occurring in some control system for a physiological system. So some parameter went through some limit, one could observe a change in the dynamics, a qualitative change in the dynamics, which may be manifest as abnormal dynamics in a person associated with disease. And we suggested that one could do experiments or do observations in which you demonstrated the onset of abnormal dynamics. You could gather and develop detailed experimental data and also clinical data to determine whether sequence of bifurcation similar to those found in mathematical models actually occur in physiological systems, and then you could attempt to devise novel therapies for disease. So in this talk, I'm going to try to give you some aspects related to this program, and I'm going to try in particular to say a little bit about the types of approaches, practical approaches that one has to actually treating disease and diagnosing disease. Okay, so I'm going to talk about mathematical and biological models, develop practical applications, and in this talk I am mostly going to talk about cardiac examples. It's what I've known the most, what we've studied the most, and also there's kind of another reason. If you go to a cardiologist and you have an abnormal rhythm, he's going to tell you what rhythm you have. He's going to do a classification of your rhythm based on qualitative properties of the rhythm. So exactly the same way of thinking that people think about in the context of mathematics in terms of qualitative properties of dynamics is really the way cardiologists think about their particular field. Okay, so this is an experimental system that we've used a lot. Let's see if I can get this to run. So this is a ball of heart cells that is approximately 1 millimeter in diameter. No, 1/10 of a millimeter in diameter. And it can beat spontaneously on its own. It's taken from the hearts of embryonic chickens. Okay? The thing that we were looking for chaotic dynamics. How do we look for it? One of the ways of looking for it is stimulating this. So this is an example of what's called a nonlinear oscillator. What does a nonlinear oscillator mean? It's not like a pendulum. It's not like a pendulum. A nonlinear oscillator is an oscillator following some pathway, but if you start off that pathway, then you return to it. Okay, so it's a stable oscillation in that you return to that oscillation. We can stimulate that with a microelectrode, injecting little current pulses into that, and you can change the amplitude and the frequency of that current pulse. As you change the amplitude and frequency, you can see many different rhythms, many different patterns of entrainment between the stimulus and the actual beating. And one of those rhythms is represented over here. So these first bumps is the actual contraction of the heart, and the spike is the stimulus. So this is experimental data. One can take the experimental data to analyze it by measuring the phase of the stimulus. The phase is just proportional to the time from the start of this contraction to the stimulus. You can plot the phase of one stimulus as a function of the phase of the preceding stimulus. So you can measure this time and plot this time as a function of this time, and then you can convert it to phases by normalizing. And for this particular trace, this is just an excerpt, you come up with this function, these points. These points are lying on pretty close to a one-dimensional line. That one-dimensional line is very close to the simple models developed by Feigenbaum and Smale and others, Jim York, other people, Robert May, who looked at what happens in these quadratic functions. And this is an example of a function that is known to give rise to chaotic dynamics. So this is going from there to there is done without any theory. It's just a representation of the data. Where does the theory come in? The theory comes in by observing that if you give a given stimulus, a single stimulus to that beating aggregate of cells, you can change the timing of the next beat. This is a phenomenon that's called phase resetting. We can measure the phase resetting function. Phase resetting, what does phase resetting mean? It means here you're in one phase of the cycle, you give a stimulus, it takes you to a new phase of the cycle. That function that represents that is something called a circle map. And by iterating the circle map, by going ahead and iterating it the same way that people who studied chaotic dynamics just in these difference equations, we can predict where there should be chaotic dynamics. So this is just experiment, but then the theory comes in terms of experimentally determining these circle maps, iterating these circle maps, and showing that the chaotic dynamics, as well as other rhythms, agrees with what is predicted theoretically. Now, at about the same, this was published in 1981. At about that time, Rafael Perez, who was the person who introduced me, so this is going back now, it's scary, right? 40 years, almost 38 years. So Rafael came and visited McGill University, and was not studying explicitly what's happening in the cardiac cells, but was looking at an extremely simple mathematical model of phase locking, of effect of periodic stimulation on a nonlinear oscillator, okay? Changing the frequency of the forcing compared to the frequency of the oscillator, changing the amplitude as a function. So this is the amplitude of the forcing. This is the period of the forcing, and observing that there are a number of different locking patterns. Now, the observation of these different locking patterns is due to the mathematical physicists, well, initially due to Poincaré, but then formalized and developed greatly by the Russian mathematical physicist V.I. Arnold, who described what happens in circle maps, that's what we're describing here, when the amplitude of when the circle map is an invertible mapping of the circle. So it's a one-to-one invertible mapping of the circle, and Arnold described what happens down here, where there's this beautiful ordering of these entrainment zones. What Raphael and I worked on is what happens when the map becomes non-invertible. And when the map becomes non-invertible, you observe all of these period doubling bifurcations, chaotic dynamics, exactly as you had in the quadratic map, except things are more complicated because in these non-invertible maps of the circle, we have both maxima and minima, and they interact with one another. So one gets quite complicated bifurcations, quite complicated dynamics, and I think that the first paper that really showed what happens for these higher amplitudes in these maps was really this paper that Raphael and I did back there. So it was extending this amazing work by Arnold to a region in which these maps become non-invertible. Now, how can we synthesize the work on the chick heart cells and the theoretical work in these simple maps? Well, we can now determine what the maps are from doing phase resetting experiments on the chicken heart cells, and then we can use the same types of mathematics that we use to compute the different zones here, which is just some iteration and a little bit of theory. And, so before I do that, we'll just say, simple mathematical models shows how phase locking, bistability, period doubling bifurcations, and CAS may result from periodic stimulation of nonlinear oscillators. That's what our experiment is doing, periodic stimulation of nonlinear oscillators. And when you try to do the application of that, which was then published in Phys Rev A in 1984, we see that we have many different rhythms that are observed experimentally. We can compute the boundaries of where we expect different locking zones. We can see where we predict chaos mathematically and where we see irregular dynamics in the experiments. So actually, there was kind of an amazing agreement between what was done based on the theory and also confirming it in experiments. And the notion here is that this type of behavior should be observed in many, many different circumstances where we have periodic stimulation of nonlinear oscillators. So there are a whole bunch of other examples. I don't think in biology there's any example that's as beautiful as this, but I'd be happy to hear if anybody knows of anything, but I'm also prejudiced. Okay, so this is one basic science which is strongly motivated by nonlinear dynamics and thinking, going back to the thing, can there be chaotic dynamics in physiological systems? And I would say the answer is yes. And there are a whole bunch of very subtle issues. Here we say irregular dynamics because Michael Guevara didn't want to say chaos because in a real system we know there are stochastic terms and there are a whole bunch of other issues happening. But we feel that we understand this reasonably well. Now, another basic science issue related to the question of dynamical diseases is something related to spiral waves in cardiac tissue. And I mentioned this paper in the first talk that I gave. I don't know how many people here have actually seen that paper or know about it. Are there many? This paper from Wiener and Rosenbluth from 1946 in the Archivos del Instituto de Cardiología de México. Norbert Wiener is the famous MIT mathematician. Arturo Rosenbluth was a physiologist at this institution, or in the Institute of Cardiology. The study was made during Dr. Wiener's stay in Mexico as a guest of the Institut Nacional de Cardiología. So what they were actually starting to do on the basis of very, very simple theoretical models was predict the possibility of spiral waves in media such as the heart. These ideas were followed up probably 20 years later by Valentin Krinsky, who was a Russian biophysicist, Art Winfrey, who was at the University of Chicago. Experimental observations of spiral waves in cardiac tissue were done initially. One of the earliest was done by Jose Jalife, who's actually a Mexican medical doctor who's now in the United States, who's done many brilliant works in physiology, Frank Witkowski, and then many others subsequently. So when we're talking about spiral waves in cardiac tissue, I want to show you what I mean. So this is an example now that's coming from cells, cardiac cells in tissue culture, which are now not a ball of cells, but are a sheet of cells. And in this, if this works, yes, there's a spontaneous initiation of a rotating spiral wave. This is about 1 centimeter in diameter. The cells from chick heart, it's imaged using a calcium-sensitive dye. And in this excerpt, you see that there's an initiation of the spiral wave, which circulates around and then dies. In this original paper that was done primarily by Gil Bubb, who was a graduate student at the time, we also tried to develop a mathematical model. And the mathematical model is really very similar to the mathematical model that was done by Wiener and Rosenbluth. The very simple idea is that you can have a cell which is excited. It can infect neighboring cells which then become excited. After a cell is excited, there's a time during which it's refractory. It can no longer get excited. So that's the basic idea. The elaborations that we put onto this is saying, well, in real stuff, everything is not homogeneous or on a grid. There are heterogeneities. And we can also have some parameter which is associated with how fast this wave can spread with the level of excitability. And then there's another parameter which is association with a increase of or decrease of excitability after it's been excited a lot, something one can just call fatigue, but is associated with the fact that if you stimulate something very rapidly, it becomes harder to stimulate. Something gets fatigued or starts wearing out. So in this, when we have very good excitability, if it works, we're saying that there are some random pacemakers. Once a wave gets started, that wave is going to go and just invade the rest of the tissue as a target pattern. And if we have a situation where there's less excitability and we have heterogeneity, then you can get targets starting, but then you start getting the initiation of spiral waves. If we also have fatigue factors, so things get harder to excite, as time evolves, you can see the velocity of propagation may slow down, the geometry changes, and in this particular case, at the end, there's actually a dying out of this rotating wave. So we have a burst of activity, and that burst of activity gradually is going to die out. Now, Gil Bubb also, as part of the work that he was doing, was try to do experiments in which he would systematically now change the density of cells, which reflects how excitable it is, and then also adds a drug which changes the communication, how well cells can communicate with one another. And as he does that, he observes a whole bunch of different situations where everything is very excitable, well connected. He sees these target patterns. As he changes the parameters, he begins getting all sorts of other rhythms. We'll see if these are running. I guess they're running. Including situations in which things appear to be very poorly organized. It can be hard to see what's happening, but you can get these little waves circulating around, which can be hard to see. So all of this occurs as the parameters in this tissue is basically changing. What we said at the time is that we believe that there's a universal organization. So this is an article in PhysRev Letters back 15 years ago, in which if things are not very excitable, we get just blocks. So a wave cannot propagate. Where things are very excitable, we get things propagating as these target patterns, and in between we can get bursts of behavior and spirals of behavior. So we believe that this should be true in any system like the heart, including your heart. It should also potentially be true. So this is an experiment, not from our group, from another group. It goes back a way. Most of the talk is about cardiac cells, but this was a paper. One of the co-authors is Stephen Schiff, who's an actual neurologist, in which they were showing the possibility of spiral waves also in the brains of rats. And one of the notions why they're interested in that is the possibility that spiral waves may be associated with some pathologies of the brain, including perhaps epilepsy. I'm not sure that people think that that's what's happening now, but it's something that has been proposed. And in other circumstances, perhaps there are also spiral waves that have been observed in certain situations, such as one called spreading depression. Now, okay, so now I'm going to switch to practical applications and talk about potential practical applications of some of these ideas. I'm going to talk about diagnosis of something called atrial fibrillation, cure of atrial fibrillation, predicting sudden cardiac death. I talked about that other days, I won't say much, and controlling abnormal rhythms and their consequences. Now, I see that there are a bunch of people. Almost certainly, at least some of the people in the room either have atrial fibrillation or know someone who has atrial fibrillation. So atrial fibrillation is typically associated with what people would call an irregular pulse. So some people have a very irregular pulse. What's the happening? Well, this is just some kind of silly thing that I got from the internet, where this was some representation of what somebody thought might be happening in atrial fibrillation. This is an electrocardiogram. These beats over here are when the heart actually contracts. And in atrial fibrillation, if you have someone who has atrial fibrillation, if you feel the pulse, it will be irregular. And if you look at the electrocardiogram, one of the manifestations is that you see irregularity in these times of contraction. So I looked up the statistics for Mexico. In Mexico, it's estimated that 426,000 people have atrial fibrillation. It increases in frequency as people get older. It can lead to stroke. So this is some statistic. I don't know where they come up with those numbers. They say about 5,000 of the strokes are potentially associated with atrial fibrillation. So this is a serious health problem. The reason why it's particularly a serious health problem is that in general atrial fibrillation is benign. It can be controlled, and people live for a long time with atrial fibrillation. So when people consult doctors with cardiac arrhythmias, the main one that they have, the main cause for doctors treating cardiac arrhythmias is atrial fibrillation, because it can last for a long time. And then these are just some observations. One of the things that's very interesting is atrial fibrillation can be paroxysmal. It may be hard to identify outside of the doctor's office. So if you go to a doctor, they may take your pulse or electrocardiogram and everything is normal, but that doesn't mean that there may not be atrial fibrillation happening another time. And if atrial fibrillation is identified, then there's medical therapy. There are drugs that will reduce the risk of stroke very significantly. Now, one of the questions, and this is a question that colleagues in the device manufacturer Medtronic presented us, was would you be able to develop an automatic way to detect atrial fibrillation? So this is a plot of intervals between heartbeats from an actual person who had atrial fibrillation. This is a normal rhythm. Notice that it's not absolutely constant. There are fluctuations in the normal timing. But during atrial fibrillation, the fluctuations in the timing between the heartbeats tends to be faster, and there's much more fluctuation than during atrial fibrillation. Notice this would be an episode of atrial fibrillation, and here's a short time when there's no atrial fibrillation in that particular patient. And this is work that was done with a Japanese postdoctoral fellow who was with me and published a long time ago, 2001. Katsumi Tateno developed a technique to analyze and an algorithm to detect atrial fibrillation. And one would say, if one's a physicist, well, just compute the standard deviation of the interbeat intervals. That was something that people at Medtronic had tried, and it didn't work very well for a variety of different reasons. What we developed was a notion of doing a histogram, not of the intervals between the beats, but the difference of intervals between two consecutive beats. So we do histograms of the difference of intervals between two consecutive beats, and it turns out that depending on what the average heartbeat rate is, these, for people in atrial fibrillation, tend to be quite consistent with other people in atrial fibrillation. And based on these probability density functions of the changes of the intervals between heartbeats, we developed an algorithm. People at Medtronic gave us a blinded data set. We applied the algorithm, and we did well enough so that they licensed the algorithm. We published the algorithm, and we patented the algorithm. And eventually, Medtronic in 2009 announced a device. The device would go underneath the skin. It would be essentially recording and recording electrocardiogram and then doing analysis. They say Medtronic, this is from 2009, Medtronic today announced the commercial availability of its revealed XT insertable cardiac monitor in the United States, along with the nation's first implant of the new device. Medtronic Reveal XT offers important insight into patients' irregular rhythms, including atrial fibrillation. So the reason why that can be important is if a doctor gives you a drug that may control atrial fibrillation and then go away, the doctor doesn't necessarily know whether that's working or not. And also, if you have some symptoms, but you go into the doctor's office, they don't know what's happening, they might want to do long-term recordings to try to identify what's actually happening. Then, a few years later, they made this much smaller. The revolutionary Reveal Link is designed to help your doctor quickly diagnose and treat irregular heartbeats that may be related to unexplained fainting. So Medtronic, of course, to implement this in their device, took some of the original idea, but then also had to do significant improvements in terms of engineering in order to get it into the device. and so forth. So they're interesting stories, but I won't tell you those now. What about competitors? So this is something which is kind of invasive. You have to put it under the skin. A couple of years ago, you saw the headline, Apple Watch. The Apple Watch can now perform an electrocardiogram, detect atrial fibrillation, and detect when the person's heart rate is too low. So now people, does anybody have an Apple Watch? Apple Watch. Anyway, the Apple Watch, as well as other devices that are becoming on the market, can now presumably detect atrial fibrillation and give you a warning. And I don't think that they are allowed. I don't think this is yet approved as a medical device, but it might say, we've noticed something and maybe you should try to check with your cardiologist, or maybe it will store some data. I've talked, I was at a meeting where people from Apple were giving presentations about this. And I said, oh, gee, that's really interesting. What algorithm are you using to detect atrial fibrillation? Obviously, I wanted to get some of the money, but they said, well, it's proprietary. We can't tell you. So I have no idea how they're detecting atrial fibrillation. It's kind of an interesting thing. From a point of view of medicine, this is an interesting development, the fact that we now have very low cost, or comparatively low cost, much lower than having a surgeon put something in here. We have comparatively low cost devices that can detect atrial fibrillation. So to diagnose atrial fibrillation, the whole field is in pretty good shape. There's a huge market, growing applications. Many people will probably have short episodes of atrial fibrillation, and it's not clear how medicine is going to be able to cope or what the treatment is. If somebody has atrial fibrillations 5 minutes once a week, they go to the doctors will have many more patients to examine, and what are they going to do with that? So there are all sorts of interesting questions which are arising now because of these improvements and new technologies. Okay, what about curing atrial fibrillation? So, well, this is a major medical problem. If you look in a medical textbook, so here's a medical textbook from 2006, Mechanisms of Atrial Fibrillation. This is written by people who know nothing about mathematics. They're very well-known, famous cardiologists. I think this is the cardiologist of Queen Elizabeth, and she's still doing pretty well. They have all of these different pictures that you can have automatic foci, you can have rotating waves, you can have rotors, just like I described before. You can have, the rotors may be stationary, they may be moving. You can have multiple foci, you can have multiple rotors, you can have very little waves, and you can have foci plus multiple waves. Well, you might recognize that all of these things correspond to all of those different rhythms that I just showed you in those experiments that we did in chick heart. So how did the cardiologists actually go about curing this? Well, there is a French cardiologist named Michel Hasseguer, who's in Bordeaux, who discovered that if you destroy certain portions of the heart near where they think there are pacemakers that are rapidly activating their heart, this is regions of the heart in the left atria near where the pulmonary veins enter the left atrium. If you destroy regions and destroy the conduction in those regions, then you can cure atrial fibrillation. So that's a procedure that was developed really in the last maybe 20 years or so. And it has, depending on the group that's doing it, depending on the report, they say it's up to potentially 80% effective in the first year, and maybe at the end of 1 year, it's less than that, maybe 50% effective. A few years ago, maybe 7 or 8, or maybe 10 years ago, a cardiologist by the name of Sanjeev Narayan suggested that you could ablate the center of foci, ablate the center of rotors. So he was saying, he was basing this on the notion that atrial fibrillation may be associated with these rotating waves. If you ablate, if you kill the center of those rotors, you may cure the person. So this is from a, this is a picture from a patient of Dr. Narayan, and it's a slide that was given to me by a cardiologist named Mohammed Shanasa. And in Mohammed Shanasa's notion that this rotor is like the eye of a hurricane, and then what Narayan was trying to do was trying to have electrodes inside patient's heart and map the center of the rotor. The kind of data that they have is shown over here. So this is meant to be from the inside of somebody's atrium, and they're trying to ablate the center of the rotor. So this represents a lot of the atrium. So they're trying to stick a catheter in the person's heart and burn near the red spot. And the claim that they made is that this is a very effective, or this can be a very effective method to cure atrial fibrillation. There was a clinical trial published in 2011, and it represented a multi-cell validation of this technique. Now, as a consequence of that, the company that was started by Sanjeev Narayan was sold. Let's see. This is a press release from 2014. Topera, developing innovative electrophysiological technologies to improve the diagnosis of treatment of atrial fibrillation. Topera has developed a novel catheter. So they also do mapping of what's happening. They try to identify rotors, which help physicians physically identify and target the specific areas of a person's heart that are perpetuating atrial fibrillation. Under the terms of the acquisition, Abbott will acquire all outstanding equity of Topera for $250 million upfront and potential future payments. So this is a lot of money, okay? If it works, great. But this paper said it didn't work. What date was this? So this was a paper that said, concluded that atrial fibrillation, that this ablation technique did not work. From a point of view of theory, if I have a rotating wave like this and I knock out the center, I'll just have something actually rotating around the rim. So from a theoretical perspective, there's not a clear logic in my mind about why this should potentially work. That paper was retracted. That's what this says. The reason why it was retracted, and the cardiologist who wrote this, a very, very well-known cardiologist, it was retracted because presumably there were some abnormalities in the patient recruitment. Subsequent to that, and now we're into 2018, there was they fixed the problems in that earlier paper, and now they say our findings did not show any benefit from the adjunctive rotor ablation in patients with non-paroxysmal atrial fibrillation. Okay, so one of the questions that we had is why are these different groups? I went to a cardiology meeting in Europe in which these two very well-known cardiologists were really almost ready to fight with each other. One of them says we see rotors, the other one says we don't see rotors. So we were wondering why is there such disparity between what different groups are saying? And what we tried to do is we tried to develop an algorithm to detect the center of a rotor in actual experimental data. And One of the co-authors of this paper is Matt Dobbs, who I don't know if anybody here knows Matt Dobbs, but he is not a physiologist. He's actually a physicist who's a cosmologist. So he's very experienced in analysis of large amounts of data and trying to see signals in data. So it was great for us to talk to Matt Dobbs at McGill. And without getting into all of the details, what we tried to do was actually systematize the analysis that was done. So I'd say there's a striking lack of analysis concerning the role of resolution, noise, and detection algorithms on rotor identification. So we wrote a paper in which we tried to do something a little bit more systematic, just to give you an idea of what the sorts of problems that arise are. So this is now tissue culture that's that's grown in the form not of a sheet of cells, but in the form of the letter theta. And the original work in our laboratory done on cardiac tissue culture, grown in different geometries, was done by Hortensia Gonzalez, who's in the biophysics department here, who's sitting over there. So Hortensia developed some of these techniques initially. And if we look and see what's happening here, this is once again the sort of irregular things that can happen. If we do some more analysis of that, then we can have color coding of different phases of the cycle. It can be very, very hard to try to figure out exactly where there might be a center of rotation. It's clear that something is happening, but are there 2 centers of rotation here? Are there 4? It's very hard to make those judgments. And this is an experimental system which is enormously higher resolution than you have in the real heart. So I would say curing atrial fibrillation, I would say more care is needed to define a rotor and to understand the consequence of ablation of its center. Drugs and ablation based on physiology empirical findings have moderate success in selecting individuals. Ablation as a procedure is unbelievably expensive, needs very expensive equipment, trained personnel, and so forth. Okay. Another practical problem is predicting the risk of sudden cardiac death. I've talked in the last 2 seminars, I've talked a little bit of some of the work that we've done. This is work that we're interested in and working on with Claudia Lermer, who's also here, who visited us in Montreal. And a recent review that we did is in Journal of Physiology in 2016. And if anybody is particularly interested in this, you could perhaps look at this article and also talk and discuss it with Claudia. Now, I wanted to finish up but just by going through very rapidly some additional therapies that have been suggested based on either nonlinear dynamics, or in which nonlinear dynamics or dynamics should be able to play a role. So, okay, one of these is Parkinsonian tremor. So people probably know people with Parkinsonian tremor. They have tremor, they can have great difficulty walking. A therapy that was developed a long time ago is to stick an electrode in the person's brain and actually doing something called deep brain stimulation. So this is rapid deep brain stimulation. Notice we're doing periodic stimulation of something which has a distinct oscillation in and of itself. It's the sort of thing we're talking about before. This is an example of a trace that was done by my colleague Anne Bouter with a neurologist named Gül, in which this is now from an actual patient when the stimulation is on. There's no tremor when the stimulation is turned off. After a few seconds, you develop this oscillation and you develop the tremor. And for people who know mathematicians, it looks like this is undergoing a supercritical Hopf bifurcation that's happening after that. We developed a mathematical model for this. The mathematical model was very simple-minded, I would say. The mathematical model was presuming that during the stimulation something was building up and decaying away. And it's actually possible to compare that mathematical model with data, clinical data collected by others, not by us, in which they test the efficacy of the deep brain stimulation as a function of the frequency and the amplitude of the stimulation. And based on that, we can set parameters for the particular model that we had. I would say that could this model be correct? Probably not, but it does show that there's a possibility of doing comparison between actual clinical data and results of mathematical model in this particular situation. To the best of my knowledge, people do not understand why deep brain stimulation works. It can be effective. Once again, it's a very invasive and expensive procedure. Now, physicists in the room may be familiar with the concept of stochastic resonance. Stochastic resonance is a notion that people developed a while ago in which you say that you can enhance detection of a weak signal potentially by adding some noise to it. For example, if you have a weak sinusoidal oscillation, which is sub-threshold for detection, if you add a little bit of random noise, that random noise may bring it above the threshold for detection sometimes. And then if you look at the signal to noise ratio as a function of the noise, it doesn't decrease with increased noise. It can once again be low for low noise and have an intermediate value of noise, which will lead to enhanced detection. So Jim Collins decided to try to improve people's balance with using these ideas by putting little vibrators in soles of shoes. And David Paterfar decided to do stimulation of newborn babies to improve respiration by having a little crib, which would give stimuli. This is a slide from Jim Collins. When he did this, he was at Boston University. Now he's at Harvard. And the notion that they have is that having little vibrating soles in shoes can enhance the people's balance and ability to balance. And the notion is that if there are elderly people who are frail and who have lots of falling, that this may improve their balance and may help them reduce the number of falls. And here was a press release from 2014. Boston researchers find vibratory stimulation applied to the sole of the foot using novel technology shows promise for fall prevention. Now, this is now respiration from a baby. This is the beginning of the respiration. This is data not collected by us. And right after that, the baby had a very long apneic spell, and such apneic spells could be associated with the baby dying, sudden infant death syndrome. So what David Paterfar is a neurologist who became fascinated with nonlinear oscillators as an undergraduate student and worked and talked to Art Winfrey and myself going back a long time ago. He's now the chairman of neurology at Dell Medical School, which is a new medical school that started in Austin, Texas. And he was proposing having vibrating mattresses, and these vibrating mattresses, according to the data that they're collecting, is helping to regularize, so this is meant to be an irregular respiratory movements in a newborn baby with big spells where they're not breathing, and then when they give the vibration, they have stable breathing restored, and then when they turn off the vibration, they come back to those abnormal patterns. So the notion, this is from 2009, but here's from 2015, in which they're saying stochastic resonance stimulation may be a non-invasive and non-pharmacologic treatment option for apnea, oxygen desaturation, and some aspects of bradycardia, slow heart rate, in premature infants. So Pedafar is very optimistic that this will actually have real clinical applications. And the ideas really have come from the physics community and these ideas of stochastic resonance. One final slide, my colleague Michael Mackey, who I mentioned right at the beginning of the talk, has remained very interested in blood diseases. In blood diseases, there are now factors that are known to stimulate and boost blood production. Generally, when hematologists give this, they have some algorithm that was developed by the drug company. The drug company develops an algorithm based on certain criteria, not necessarily the optimal criteria. By doing mathematical models of drug development, Mike Mackey and his colleagues were proposing that one could give a little bit less of these very, very expensive biologic drugs and still get very good efficacy. And that Michael is now working with groups of people who are making suggestions, I think very practical and good suggestions, for how to continue to improve the therapy of this based on mathematical models of these blood cell production systems. Now, to finish up, in terms of implications, there's been a large body of research that has studied bifurcations in biological systems as a function of parameters of the system, and such parameters can involve time delays, stimulation frequency, structure of the system, and also drugs. So this is still a fascinating range for model systems where one is able to do manipulations. In terms of diagnostics, there's great interest in studying dynamics of physiological system. I would say most of the interest is maybe for somebody who's riding his bike up a hill to actually have a printout and say, oh, look how I'm doing. But from a medical point of view, you now have the ability to collect vast amounts of data, which has enormous potential, I believe, in medical treatment. And exactly how that data will be collected, will be coordinated and so forth is completely open at the moment. But the development of wearable devices and public databases of data has potentially enormous potential implications for health, which are all in the future. In terms of therapy, use interventions to restore normal dynamics using surgery, stimulation, and drugs. The developing of good therapies is something which is still very hard, and there are isolated examples, perhaps, where it's working, but it's hard to do that. I once again acknowledge, great benefits I've had from collaboration with many colleagues, students, postdoctoral fellows along the years, including especially colleagues in Mexico. So thanks very much for coming, and I'm happy to answer questions.
We have time for some questions.
Yeah.
Well, when you have an apparatus and you say that you are measuring something, and is this apparatus has to be certified by some agency of the state? And if you don't know the algorithm, how there are problems on that, or you can have a company say, well, this is what I'm saying I'm measuring, and this is going to be good for you.
Okay, so I would think that there are problems with it. The people at FDA, and I don't know what the situation in Mexico, I'm not sure they need to know the algorithm. One of the things that I saw about a week ago or 2 weeks ago is there was a cardiologist in New York City who had a patent for detecting atrial fibrillation, who said he's suing Apple for $2.1 million or $20 million for patent infringement. Now, I looked up his patent, and it didn't seem to me that it was necessarily going to be a really good patent, but Apple might be happy to pay him $2 million because that's nothing to them. So There are a number of patents for, I didn't exactly tell the truth about our patent. Our patent is licensed to Medtronic. Medtronic gave a payment to McGill, and I got a little bit, I got a share, and I will only get more if they sub-license it to somebody else. So now if they sub-license that algorithm to Apple, it's perfectly possible. I don't expect that company will send me a letter saying, "Dear Leon, we're happy to give you another bunch of money because we've sub-licensed that." But I think it's a very strange situation where they won't tell, and I communicated with a few people, including their engineering team, and they said, well, they sent me something, but the thing they sent had no information.
More questions?
Leon, can you tell us something about the ventricular fibrillation predictions?
Oh, so the predictions of the sudden cardiac death. Okay, so at the moment, I'm not sure, there's a device called an implantable cardioverter defibrillator, which, if it detects ventricular fibrillation, will give a shock to the heart, and in most cases will convert that rhythm. The criteria to get that implanted now are, one, having suffered a cardiac arrest and not died from it, being recovered, and also if you have poor ventricular function called a low ejection fraction. Okay? In other words, the ventricle is not pumping very efficiently. Those are the two main criteria, according to my understanding, to implant implantable cardioverter defibrillator. A very large number of people now who get the implantable cardioverter defibrillators, it never fires. They never get a rhythm that requires it to fire. And of the people who do not meet the criteria, a large percentage of those, not a large percentage, a small percentage, but a small percentage of a large number of people suffer sudden cardiac death. So there's not good criteria to analyze the risks for sudden cardiac death. There are a very large number of clinical studies. Most of these things will do things where they measure particularly easy things in the electrocardiogram to measure, such as the standard deviation of the interbeat intervals. Or more sophisticated, they may measure a scaling coefficient of a low-frequency power spectrum of the heart. There may be I have a slide someplace here where there may be 20 different of these non-invasive measures. Each one of them has some predictive value, but none of them have a good enough predictive value to make a judgment. In the work, so these are all based on large clinical studies of hundreds, if not thousands of people, where you measure all of these people, you see who dies, and you try to see, did they have a power spectrum with these characteristics? What I'm working on with Claudia is trying to look at individual cases. We know that many people have extra heartbeats called premature ventricular complexes. It's very common, but they're very rich dynamics that are present during those rhythms. Most cardiologists do not look at the details of those very rich dynamics. We are interested in trying to understand the mechanisms associated with generating those rhythms based on our understanding of nonlinear dynamics, in some cases probably associated with two competing oscillators, in other situations maybe with some kind of reentrant rhythm. So there are only a few mechanisms that one can imagine, but the dynamics that you can observe are very rich, very complicated, and very frustrating from my perspective to do, because it's very hard to understand what's happening in an individual patient. I believe that looking at N as one patient, is a perfectly reasonable enterprise to do. Certainly if you're a physicist, you can publish the spectroscopy. of a given substance or the molecular orbital of benzene. So you do N is 1, a benzene molecule, and that can be a good paper. In cardiology, the style of looking and doing detailed analysis of one patient may be a case report, but generally doesn't have much weight. So the strategy we're taking is trying to understand what's happening in detail in individual patients. And whether it's hard to do, whether we'll be able to classify the patterns, and whether it will have clinical impact is something we don't know yet.
More question?
In the science paper, you showed why was your name second?
Why was my name second? Oh my goodness. So the actual model for the hearts, for the blood cells, was developed by Michael Mackey. I did a model for the breathing. So I think that's probably the main reason. Michael had been there, was a little bit more senior to me at that point. So it's not, I think that was probably a fair way of doing things. So.
Well, I just missed something in these spiral waves.
Yeah.
They are waves of what? They are polarization waves, or which kind of waves?
Oh, it's waves of contraction, really.
Or contraction of the muscle?
Yeah, contraction and depolarization. So there's really a wave of contraction that goes around.
The detection is to what?
Okay, it can be done in different ways. What I showed was calcium-sensitive dyes. It can also be done with voltage-sensitive dyes. And Gil Bubb is now just using it from the motion artifact. So he subtracts one frame from the next frame, and then he just can visualize all of these complex dynamics.
And the ablation means what? You burn?
Yeah.
The center, actually?
Yeah, you do. You fry it. The cardiologists have an expression called learning while burning.
Okay. Another question?
You just told us that it's frustrating and difficult to understand the nonlinear dynamics in the heart. Can you tell us an experience, a particular experience, when you felt like frustrated because you were not understanding something about it?
Sure, I did that in the lecture yesterday. I showed data from a case that we were looking at and talking to Raphael about, where it's clear that you're seeing rhythms that are not random. They have great structure. Mind you, we haven't tried to do detailed modeling yet, but since we've worked with models related to circle maps, we know what should, have an idea without doing all the computations, and it didn't seem like the simplest way we thought that it should be modeled would actually work. Now that could just be because we haven't really sat down and done it yet, or it may be because we're missing something. In the real heart, in a lot of these models, there are always some parameters. The simplest model is there's the normal heart rhythm and there's an abnormal rhythm competing with it. The simplest model is both of these rhythms are absolutely constant, and the refractory period is also constant. We understand everything that happens. I think that's probably true. We understand almost everything that happens with that model. But any of those parameters can change as a function of time. And if those parameters change as a function of time, we don't have control of that. We have no way to measure that if we're just looking at the electrocardiogram. Well, for the normal heart rhythm, we can see what that is, so we measure that directly on the electrocardiogram. But if this abnormal rhythm is changing as a function of time, we have no way to directly measure that. If the coupling is changing as a function of time, we have no way to measure that. So one can try to infer those, and that's the sort of thing that we try to do.